Okay, we're back to cell division cycle video 2. Refresh. We're talking about the cell divisional cycle, which is composed of inner phase and the M phase. Inner phase has the G1, the S, and the G2 phase. We said the G1 phase was the immediate time that the two daughter cells started their time clock. The immediate time the two daughter cells started their time clock. We said that the G1 phase is a growth phase and also the time for differentiation to occur, for the two daughter cells to grow and get back to getting all the organelles and other chemicals needed for there to be the two liver cells and two kidney cells. The S phase, which was around 10 to 12 hours, was for duplication of DNA. So where we stopped was at the G2 phase. The G2 phase, as I alluded to, was the time phase right before you started the actual M phase. The M phase. The G2 period is the time where you're getting totally prepared to have everything necessary to go through the M phase. The M phase is the actual mitosis. That is the only phase that you can see under the light microscope, which is the M phase, because that's the time in which the genetic material is called into the chromosome form. So here's the M phase. You can see it only lasts 1 hour. It's the shortest. It's the shortest. It's the actual event. It's the stage play. We will be talking about mitosis in a separate video. Its various phases. It has several phases. But this is the time period there. So we go further, because we'll do that in a separate video. So we go back then. Now I'm going to talk on the checkpoints. Remember what I said, that a cell, when it needs to function, must leave the cell divisional cycle and go into G0. Leave the reproductive aspect alone and go into G0. That's the time in which the cell does its work as a kidney, liver, spleen, or whatever. All right. There are checkpoints where the cell will leave or check itself. At the end of G1 is checkpoint 1, known as the restriction point. This is the major one. Now listen to me. This is the major one. This is the one that gets all screwed up in cancer, because the cell no longer has this and keeps going and doing this and making a bunch of cells that do absolutely nothing, just crowding out the good cells. So at the end of G1 is checkpoint 1. Now in another video, I'm going to loosely describe the biochemicals involved in these checkpoints, but not very detailed, even if you were face to face. Once you get past this, the cell then goes through S and G2. Why is the most likely spot to have the checkpoint at the end of G1? Now remember, in a cell, in the nucleus, we said the nucleus was about 5 millionths of a meter, but you needed to put 2 meters of DNA in there in a somatic cell. 2 meters in 5 millionths of a meter. Now let's think about that. Why then, it's already a crowded nucleus. Why would you do S phase, duplicate to 4 meters, and you weren't going to even divide for a while and crowd up the nucleus? So the best spot to have it is here before you crowd up the nucleus with more DNA that you're not going to use. So if the cell is not going to do anything, then checkpoint it here. If it's not going to divide, exit the clock and go into G0. Then if you're going to come back to divide again, come from G0, come back into the clock right here, and then finish the job out. Now here's checkpoint 2. Checkpoint 2 is more or less, like I said, on a stage play. Stage director says, wait, everybody stop. Do you have everything? We're getting ready to go on stage. This play is going to be 2 hours. We're going to have to move fast. We don't want to disappoint the audience. So right here, checkpoint 2 is to be sure that all the chemicals and all the organelles are ready before you actually go into this 1 hour period of mitosis. Okay, that's kind of the logic behind it. So here again is this, showing you another one. Here are the checkpoints you can read. Why do these controls ensure cell division? Make sure each phase occurs in its proper order. Make sure the actions are properly completed before the cells moves to the next one. So you can read what I wrote there. Restriction point, G1. You can read. I kind of described it, but you're able to read it. Here again is that G1 checkpoint. There is one in M2, another checkpoint, kind of a minor one. Now this is the only one that the cell will actually leave the cell divisional cycle. These two here just slow it up. These two here just slow it up some. This is to say, are you ready? This occurs while you're in the M phase, just to be sure all the steps are occurring in a sequential fashion. Here's kind of a little picture to show. You know, G0, if you're not ready to go into that S phase and not going to divide ultimately, go into G0. G1 check, G2 checkpoint, locating the energy, triggering the start of the M phase. Okay. M checkpoint is locating it allows one internal stage to proceed to another within mitosis. That means it's just being sure when we get to the M phase that all the steps are going in a sequential order. Hopefully this kind of explains to you the cell divisional cycle. An important aspect, you can't understand tumors, which I'll have a video on that, unless you kind of have an understanding of what. Remember, you can't understand what goes wrong until you understand what goes right. Remember, all of you that are taking the course, pretty much 99% are going into some clinical field. Dental hygiene, nursing, respiratory therapy, a whole plethora. Sad as it might be, if people didn't get sick, we wouldn't need healthcare workers. So all we deal with, as a physician myself, are people that are trying to stay well, which is the new push called wellness, as you know. The old way was, don't see the person till they're sick. But in either case, people never got sick. But sickness means something going wrong. How do you know what went wrong until you know what goes right? So always keep that in your mind as you pursue your clinical field. Thank you.